So, yesterday we were talking about how one molecule, one epinephrine combined, activates G proteins. Guys, shh. Devin. All right, activates this enzyme, adenylyl cyclase, which then converts ATP to cyclic AMP, Roman, and then activates protein kinase A, which then activates another kinase, which activates another enzyme, which eventually triggers glycogen to be broken down. Remember, glycogen is long chains of glucose molecules, and so it breaks down into glucose, and that glucose can be used for energy. Epinephrine is an example of a hormone that's released. Have you guys heard of a fight or flight response?
Yes.
All right. Epinephrine is a hormone that is released in that response. We'll talk more about it later on in the course, but and triggers your liver cells to release more glucose, and therefore the glucose can get to your cells, which then can do cellular respiration, which then makes more ATP for you to have energy to either fight or a dangerous, like if you see a bear, all right, in the woods, all right, and you need some energy to, hopefully you don't choose to fight it, but if you do, or run, all right, either one. And so epinephrine is this molecule that does that. And so, I think, did we talk about these? No, we literally didn't get past this. All right.
No.
All right, so then many other signaling pathways regulate the synthesis of enzymes or other proteins, usually by turning genes off or on in the nucleus. So the final activated molecule may function as what we call a transcription factor. And a transcription factor turns a gene on so it can make a protein. So it turns a gene on so it can make a protein. That's what it means. So, going back to this picture here, all right, notice that the binding of the signal molecule triggers this cascade of this phosphorylation cascade, and eventually you get to the end here where it converts an inactive enzyme to an active form. So what does this mean? This inactive enzyme is already in the cell. It's floating around in the cell, but it can't be used because it's not in the right shape. And so therefore it doesn't become active until the signal molecule binds. All of these reactions occur and it activates this enzyme. And now the reaction that this enzyme catalyzes, the conversion of glycogen into glucose, can happen. So what this is showing you is that sometimes in the cell you don't have the protein to be activated. There's not any protein, no enzyme in there. It hasn't even been made yet. And so that's what this picture here is showing you. So this picture shows you, here's your hormone. An example of a hormone is growth factors. I'll talk about that in just a second here. Binds to a receptor. The receptor is activated usually by changing shape and triggers a phosphorylation cascade. And notice at the end of the phosphorylation cascade, so that's the transduction part, this end molecule that becomes activated goes into the nucleus. So this molecule, this is an activated enzyme, goes into the nucleus, all right, goes into the nucleus. That's what we see here. And what happens in the nucleus? Notice here that there's what we call an inactive transcription factor, and this enzyme activates it by doing what to it? What does it look like it did to it? Inactive to active. It changes shape and added a phosphate here. So this phosphate, so this is it's active because a phosphate is added, and this enzyme helps that conversion happen. Phosphate is added and it changes shape. So what does that mean? That means that it's going to bind to a gene, And this gene, notice here it says gene to mRNA. Here's the nuclear membrane here. If I were to draw the rest of the nucleus, so here's the rest of the nucleus. What happens is this nucleus has the pores. That's what this part here is. And so the mRNA leaves and goes to the ribosome. The ribosome is where proteins are made. And so remember, the DNA never leaves the nucleus, so mRNA is, the instructions from the gene is copied on mRNA. mRNA goes to the ribosome, and at the ribosome is where your protein will be made, and this protein will lead to your response. And so what we just wrote is that, that sometimes this protein that's going to elicit your response isn't made. So notice here in the cell, before I wrote all this stuff here, before this molecule binds to it, this gene isn't turned on and the cell doesn't have this protein to make the response, to give you the response. So what happens is when your signal molecule binds, the end of the phosphorylation cascade triggers a gene to be turned on, which then makes the protein to give you a response. So sometimes the protein's in there is inactive and doesn't working until the signal molecule is there, and sometimes the protein's not even made until the signal molecule.
Is that still a response then, or would that be transduction?
What is what?
The protein, no, not protein,
This is still the, like all of this, turning on the gene, and therefore it's considered the response. Yeah. So like all of this would be, yes, the response. The response. I guess I should say the protein will do a certain job in the cell, but the response is turning on the gene, which causes that to happen. So, all right, that's all collectively the response. All right, and so so then looking at these pathways, these multi-step pathways have two important benefits: amplifying the signal and thus the response, and contributing to the specificity of the response. So I'm going to talk a little bit about amplifying the signal. So, so I'm going to stop here in the notes, and because I want to go over the signal transduction packet that you did yesterday, because it shows you some examples of amplification. And so then we'll come back to the notes here. So I'm going to put up here a picture here as we go over the answers, so we can look at the picture here together. So there's three different models or pictures that the sheet asks you about. And so number one on here, it says, does the signal transduction pathway occur inside or outside the cell? So here's the cell membrane, so it occurs inside the cell. And it asks you to compare the shape of the ligand to the receptor. So you should have that their shape is complementary, that the ligand fits into kind of like the active site of the receptor protein, that there's a spot that it fits into nicely. And then number three asks you to label, and it says the 4 steps in the signal transduction pathway. Our book only talks about 3 steps. What's the 4th one that they added here? Signaling. All right, signaling was not, we had reception, transduction, and response. And so in this case here, they added signaling. So this means that the signal molecule comes from another cell. So this is the ligand coming from a different cell being made somewhere else. And so that is step 1. Then step 2 is, I'm just making sure I'm in the video here. 2 is the receptor protein, where the reception takes place. The ligand binds to the receptor protein, so that is step 2. Then transduction is this whole part right here. So this is step 3, the transduction. So this molecule, the receptor, usually this receptor is activated by changing shape. So it changes shape and then activates the relay protein, and this is activating multiple proteins, and you get your response. So this is step 4, your response. Okay, so then number 4 asks you, propose a definition for transduction based upon what's happening here. And so a definition for transduction is that the signal gets moved through the cell to the spot kind of where the response is going to occur. So the signal is transduced or moved through the cell, and it changes shape of the proteins as it goes along to eventually elicit your response. 5, it pulls in this amplification, which we just talked about. So signals become amplified. And so A asks you to define what amplification means. So amplification means to make stronger, to make larger, and so on. And so in this picture of the signal transduction pathway, where's an indication of the signal becoming amplified in this picture? Roman?
When activating the relay protein 1 is activated and 3 arrows go out to 3 different proteins.
Yeah, protein, activated relay protein 1. There's one of these molecules, but notice this one molecule activates 3 relay protein 2s. All right, so that's the 3 of them. So what's happening is amplification. That's what happens in amplification. One molecule doesn't activate just one other molecule, activates many molecules. And so over time, like you can imagine that, let me draw down here, if you have one molecule that becomes activated, and that molecule activates 3 more molecules, and then each one of these molecules that become activate 3 more molecules, and these 3 at the end elicit a response from that one molecule. Because each step is amplified, you get a huge response as a result of just one molecule. So that's the benefit of that. And so that's what actually what was it? Nope, I thought there was a question that asked you why that is there, but I guess not. All right, so then 6 asks you to list several possible responses. So at the end, you can get responses. And what are several responses? And it told you that it gave you some examples in the Y box, which is at the front there. So you can produce a protein that wasn't produced before, turn a gene on, which then ultimately will produce a protein. Protein channel can be opened and then things can diffuse or move that wouldn't be able to as before. A cell can trigger the response, the cell to divide, to grow and then divide and make more cells. And then at the end of the hour today, we'll talk about that it actually can trigger cell death. So the cell can actually die as a result of this as well. And so for 7, once the response is achieved, so once you get the response, what would need to occur to stop the response? So to stop the response, actually a couple of things. You need to deactivate these relay proteins. So usually deactivating the proteins, how they become activated in the first place, this picture isn't showing you that, but you guys remember how these proteins become activated? You add a phosphate group attached to them, and that activates them. And so therefore you need to deactivate them, and that's usually by dephosphorylating or taking off a phosphate group. And then also one other thing is the signal molecule has to come off too. And usually the signal molecule at the very beginning doesn't stay on forever, so it attaches and triggers the first relay protein to be activated, and then that signal molecule comes off and the rest of it happens inside the cell. Okay, so that is model 1. So then model 2, let me erase my stuff from last hour here. So here's model 2, and the first question asked you for this is describe the event that sets off the phosphorylation cascade. So that event you should have is the reception here. So the ligand binds to the receptor molecule and that triggers that. And so then 9 asks you about phosphorylation. So molecules become activated by being phosphorylated. And so A asks you which step in the phosphorylation cascade illustrated in this model include phosphorylation. So let's just go through this. So here's the ligand that binds and changes the shape of the receptor. The receptor activates the relay protein. So you have this active relay protein, but notice that there's no phosphate attached to it. So therefore, it wasn't activated by phosphorylation. So 2 is not an example of phosphorylation. Then this active relay protein, what does it do? Is it activates protein kinase 1. Did protein kinase 1 get phosphorylated? No, there's no phosphate group attached to it. So then we go, this protein now that's activated activates the protein kinase 2. Now notice here, protein kinase 2 from the inactive to the active state becomes phosphorylated, and that phosphate group came off of an ATP. And so therefore, step 4 shows you phosphorylation. So protein kinase 2 is active because it was phosphorylated. Then this enzyme converts inactive protein kinase 3 to active protein kinase 3 by adding a phosphate group. Where did that phosphate group come from? From ATP converting to ADP. So step 5 is a phosphorylation step. If we look at step 6, what step 6 is just showing you is that these active protein kinase 3 goes into the nucleus. And that's all that's going on there. So protein kinase 3, nothing has really happened in terms of phosphorylation. It still has its phosphate group attached to it. But what happens in step number 7 is this protein kinase 3 takes inactive transcription factor and makes it active. How does it do that? By adding a phosphate group, and that phosphate group came from ATP. And so step 7 is showing phosphorylation. So step 4, 5, and 7 is our answer there. And B, I've already answered a couple of times. Where does that phosphate group come from? It comes from ATP being converted to ADP. Okay? And then 10, the enzymes that do this, that add phosphate groups, are collectively called protein kinases. And so that's what those are. So then 11 asks you to identify the different parts of signal, or the parts of the cell signaling. So reception. So reception is right here at step 1. So that's it for reception. For transduction goes from 2 to 6. So that's transduction. It's 2 to 6. And then in this case here, step 7 is the response. The response is kind of turning on a gene. So therefore, the active transcription factor binds to it, and therefore the gene is quote unquote working. What does a working gene mean? It means that gene, remember, a gene is a section of your DNA, and that working gene will now be able to produce a protein. That protein could be an enzyme that will catalyze a reaction inside the cell, and that protein would never be made if that ligand wouldn't have bound to that, all right, to that receptor. All right, so then number 12 asks you, what is the cell's response to the signal? So the response is the transcription of of a segment of DNA, or turning a gene on so it's able to make a protein. That would be another way to say that, is to turn on a gene so it's able to make the protein. 13 says steps 3, 4, and 5 are shown as amplification steps. Describe what that means in terms of the signal transduction pathway. So if 3, 4, and 5 are amplification steps, what does that mean? It means that step 3 here, where this active relay protein activates a protein kinase A, this active relay protein isn't going to just activate one protein kinase, it's going to activate many. All right, so what it does is allow each kinase molecule in step 3, 4, and 5 to activate more than one of the next enzyme. And so therefore you get the snowball effect where you get lots and lots of molecules activated, just like I kind of showed you in that last picture when I drew the circles on the bottom of the page. So 14 asks you, what is the advantage of amplification? So the advantage of amplification is that you can get from one signal molecule many responses. And so you get a large response from one signal molecule through the amplification process. So then 15, what would occur in the cell if the activated protein kinase enzymes continue to be active for a long time? So in this example, this active transcription factor would stay active, so the gene would keep on working, making more and more proteins, and so therefore you continually get your response all the time. So 16 asks you what you would need to do to deactivate the protein kinase enzymes so you don't get your response all the time. You have to take what off of it? Phosphate groups. All right, so you take off the phosphate groups and it deactivates them. So 17 asks you to draw in some things on here that shows how you would deactivate your protein kinases and using these enzymes called protein phosphatases. So looking here, let's look at step 4. You have active protein kinase 2. So it became active by adding a phosphate group. So we could add an arrow here taking the active protein kinase to becoming inactive. What does that is the protein phosphatase, and how it does that is it takes off a phosphate group. So a phosphate group would come off and it becomes inactive again, and that enzyme helps with that. Same thing goes with this one. So this one's active, it becomes inactive. So how does it do that? With the help of protein phosphatase, and loses a phosphate group. And so that's how we could add that into that. 18. The 18 is kind of the same kind of question that we went over yesterday with cell communication, talking about how these guys, this can be used as evidence for evolutionary relatedness. And again, if you're looking at different organisms, if the organisms use the same system, so they have the same ligand, the same receptor, the same collection of proteins that are activated, and therefore the same gene that's turned on and the same protein that's produced, that indicates relatedness between the organisms. All these things are made due to the DNA, and so therefore DNA is similar between those organisms, and therefore they're more closely related than organisms that would not have the same signaling system. Okay. All right, and the last picture is with secondary messengers. In our notes, we talked about 2 secondary messengers. We talked about cyclic AMP and calcium as secondary messengers. This shows you a little bit different situation. It says, consider the signal transduction pathway. What event begins the process of producing a cellular response? So again, the ligand binding to the receptor right here. Then 20 asks you to describe the type of diffusion that is occurring to get these secondary messengers into the cell. So in looking at this and what we know about ligands and what happens when they bind, you should be able to look at this picture and say, okay, the ligand is binding to the receptor. The receptor is going to change shape and activate this relay protein. This active relay protein is going to come over here and attach to this transport protein. And then by the looks of it, then allows the secondary messengers in. How do you suppose that the attaching of this active relay protein allows the secondary messengers in? What do you think it does to the transport protein? What would you hypothesize? Yeah, it changes shape, and so when it changes shape, it allows the secondary messengers to come in. The secondary messengers are not going through the lipid bilayer, they're going through a transport protein. What's the name of the type of diffusion that you need a protein for? So it starts with an F. Facilitated diffusion. So the molecules go in through facilitated diffusion. It tells you in the stem of the question that it's diffusion. So therefore, where would you assume then that the higher concentration of the messenger molecules are? Outside the cell or inside the cell? outside the cell because they go, diffusion doesn't require energy, meaning that things go from a high to a low concentration, and so the secondary messengers will go into the cell once that channel is open and it's allowed to do that. So then B at the bottom, 20, what activates or opens the protein channel that allows the messengers to enter the cell? The attachment of the active relay protein, all right? So it could change the shape of the transport protein, allowing the secondary messengers in, okay? And then the secondary messengers activate the first relay protein, and it shows you one here, but you could have a signal transduction pathway and eventually lead to a response. All right, and then the very last question asks you, within an organism, it's critical that signals between cells are very specific. For example, if ligand A is meant to activate immune system cells to reproduce in response to an infection, it should not also cause other cells to grow as if they had received a growth hormone. When a ligand is released, what prevents all the cells in the body from being affected? Who wants to take that one? What prevents all the cells when a ligand is released? How does it only affect the immune system cells and not trigger other cells to grow? Nathan?
Because that ligand has like a specific receptor that only the cells in the immune system would have, and so no other cell except for the immune system cells would receive it and trigger a response.
Absolutely, absolutely. So the cells have to have the correct receptor. All right, so let's look at going with this. Let's go back to this here. I have a question for you right here. This isn't in your notes, but I just want to kind of goes with the same idea here that we just finished up with. Epinephrine, which is a hormone, acts on both heart and liver cells, yet the responses are different. So the example we just looked at here is that, as Nathan said, that if the immune system releases a signal, and the signal is only supposed to go to immune system cells, those immune system cells have receptors and other body cells don't have those receptors, and that's why communication isn't happening. So in this case here, we have the same ligand, the same hormone, epinephrine. It's communicating with two very different types of cells in our body. Heart cells and liver cells are very different. So the same molecule communicates with those cells, but the response in those cells are different. So what I'd like you to do is to talk a minute with the person you're sitting next to and come up with how these cells must be similar and different for this to happen. So what's allowing that to happen? Similarities and differences between
So the transduction pathway, same molecule. Oh yeah.
Let's look at what could be a similarity between heart and liver. Joe?
The receptor site is the same for both cells, considering the epinephrine castifies both.
All right, yeah, so we have the receptors in the membrane of the heart cell and the liver cell doing the same, the receptor site. So therefore they have the same shape, therefore the epinephrine can bind to both types of cells, all right? And really what's the important part of the shape of this receptor? The part that the active site where it needs to be the same. So do you think you could have kind of a different shaped protein but still have part of it the same? All right, and if the active site is the same, then they can bind to it. So so they don't have to necessarily be the exact same receptor. And so that active site just needs to be able to bind to epinephrine. So that's how they both can react to epinephrine, but what happens as a result of that is that in the heart, we get, I'll just say response A, and in the liver, we get response B. And so how do we get different responses from the same molecule? And there's a couple of different things that can be going on here. Shane?
So I was thinking that it's kind of like the HIV thing where you have like a what is it called again? You know, where it's like the additional thing. Like,
Oh,
you
a
don't-
co-receptor?
Yeah, but like you don't need it, but it just changes the like action, I guess. So it's like you need it, but it's different for the liver and it's different for the heart.
What's different for the liver?
The co-receptor.
So you're saying here that when the epinephrine binds, so instead of drawing it like this, you kind of have two receptors
Yeah.
like this, and then the epinephrine somehow binds to, both of them? Is that what you mean?
Yeah.
All right.
But
And-
the other co-receptor is different for the heart. That's what
Okay.
it is, but-
Okay, so this co-receptor over here would be different, so maybe, do you mean like,
So like in terms of colors, so it'd be blue and red on the liver and blue and green on the heart.
okay, so they have one in common, and then the other one, okay, nice. Max, you had your hand raised.
Yeah, I was gonna say that the transduction pathways could be different. So the signaling could activate different proteins in the heart and the liver, which would activate different second proteins, and that would elicit a different response.
Okay, yeah, so in the signal transduction pathway, it activates this green molecule, which then activates a blue one and keeps on going and going and going, which activates a different molecule, which will give you response A. Is that what you mean?
Yeah.
All right, and so over here, it could activate another molecule, I'll put it as orange there, and then the orange can activate the, let's say, pink, and so on and so forth until the activated molecule gives a different response. Is that what you mean? Okay, absolutely. So you can have a different collection of proteins inside that will elicit a different response, these signal transduction pathways, even though you may have the same exact receptor. Anybody else have a different idea? There's one other one that's a possibility, is that instead of at the end of the pathway here, before you get to the response, the molecule, it's kind of like one of the last pictures we looked at, the molecule could go into the nucleus and turn on a particular gene, all right, which then would make a protein that might give a response that's different than this one if the protein enters and turns on a different gene, all right, that would elicit a different response as well. So it's another possibility of that, all right? And so I just want to point out that not only the receptors and whether or not the cells have the receptors make a difference, but also the collection of proteins within the cell can make a difference in the cell's response. All right, so that leads up to the specificity of cell signaling. So different kinds of cells have different collections of proteins. So we're in our notes now. All right, different kinds of cells have different collections of proteins. These differences in proteins give each kind of cell specificity in detecting and responding. So what proteins are involved in detecting a signal? Yeah, so this is the receptors, right? So these are the receptor proteins. And it says specificity in responding to a signal. What kind of proteins are we talking about that would be involved in responding to the signal? Proteins in the inside the cell in the signal transduction pathway here. So these would be proteins in signal transduction pathway. So that response depends on those collection of proteins within the cell and which ones are going to be activated, and that will give you your response. All right, ultimately. So then it says pathway branching and crosstalk further help the cells coordinate incoming signals. So it gives you, let's look at this picture here that gives you 4 different examples of 4 different types of cells that are eliciting different responses, sometimes from the same signal. So what you're looking at is 4 cells. Cell A, look at here, cell B, cell C, and cell D. And notice here, they're all response 1, and this cell is different than the response 2 and 3. What that means is these are different responses, and they're different from the response in this cell, and response 4 and 5. So let's look at this. So a signal molecule binds to the receptor, activates the proteins, and you get this phosphorylation cascade, and eventually giving you your response. So the pathway leads to a single response, one response there. Cell B, notice cell B, the shape of this ligand is important. It's the same ligand and the same receptor. So this cell now has also binds to this, the ligand binds to the cell just like in cell A, but we don't get the same responses. Why? Are the collection of proteins inside the same? No, they're supposed to look different. All right, so they made this little circle smaller than this one in different shapes and so on. So the proteins that are activated here are different. So therefore, we have, in this case here, this activated protein activates 2 different molecules, which you get 2 different responses here. This is kind of like amplification. One molecule is activating 2. All right, and so you get 2 different responses here from the same molecule. And so that sometimes happens where one ligand produces multiple responses. Cell C says crosstalk occurs between 2 pathways. So here's one pathway, here's another pathway. Notice that the last part of both pathways are the same, so you end up with a response. So here's the same molecule, the same ligand here, the red triangle here with the same receptor, activates this molecule, which activates this molecule, which gives you a response. Notice that these guys are different than the proteins up here, so that's why the response 4 is different than 1, 2, or 3. This is another whole signal molecule right here binding to a receptor protein. Activates a different molecule, which then activates that same molecule, which then can lead to the response. Notice that this says activation or inhibition. So this molecule here can either be a molecule that as it goes through this pathway activates this molecule and triggers the response, or sometimes you can have another molecule and another hormone, let's say, that binds, and what it does, instead of activating, triggers the activation of this molecule. It triggers the deactivation, and the response will stop. So these guys can sometimes work against one another. And so for the response to happen, you'd have to have maybe more of these molecules. And then lastly, cell D is we can have different receptors. So that's what they're showing you here, that we kind of talked about with these receptors here, that the active site is the same. So the same ligand that came up to cell A and B can bind to this also receptor, but this receptor is a different shape. And therefore activates a collection of different proteins, which gives you a different response. All right, and so that's just 4 different cells showing you how the responses can be different based upon not only the receptors but also the molecules within them. All right, and then lastly, eventually you want to terminate or stop the signal, and so you don't want the response inside the cell to go on forever. Inactivation mechanisms are an essential aspect of cell signaling. When signal molecules leave the receptor reverts to its inactive state. All right, and so you have a picture there that says scaffolding or something like that. You don't need to know that that picture. It's kind of right before the termination. I forgot to delete it from the thing. Okay. There's a few other things that I want to relate to chapter 11. So on the very back of your packet, you have a blank sheet. You're going to write some things down. The thing I want to relate to this is the idea that cells can produce a ligand that induces them to die. All right, and so this process of kind of like cell suicide is called apoptosis. And so I want to talk a little bit about that and how it relates to that. So where it says concept 11.5, you guys don't have an 11.5 in our textbook, but the newest textbook, the newest edition of our textbook has an 11.5. And so yesterday I gave you a loose leaf sheet of paper. That's the copy of 11.5 from the newest textbook. That's the actual reading for it. Okay, so that's what you got there. So you have that. So I'll give you a minute. I'll have you write it down, and then I'll discuss it. So it says this apoptosis can be triggered by It can be triggered by when there's things wrong with the cell, so DNA damage in the nucleus. So you don't want the cell to divide if there's something wrong with it and make more cells that there's something wrong with it. Or if the ER is not working properly and it's misfolding proteins, so proteins don't have the right shape, so then they're not going to be able to do the job. And so there's going to be some issues with the cell because of that. And then how this relates to this chapter is the extracellular death signaling ligand. So what happens is that the cell here has receptors in it, and it can make its own ligands, and then this ligand can bind to its own receptor, so it's communicating with itself. That was like the autocrine signaling that we talked about yesterday. And this signal, this death signal, what that does is trigger a signal transduction pathway. So then that initiates a signal transduction pathway that activates enzymes, and these enzymes will start to break down the cell. And so therefore the cell, all the organelles and things like that will start to be, as we wrote up here, chopped up, and they're packaged into vesicles. And then other cells surrounding it kind of engulf them through endocytosis and digest and get rid of the cell. So the cell doesn't just kind of explode and exude all its contents around to the other existing cells around it. And so the enzymes in the cells don't digest other cells around it because you just want to destroy that particular cell. All right, and so that's what happens. And so there's 3 more bullets here that I want you to write down. All right, so, and then we'll be done. And so apoptosis evolved early in animal evolution and is essential for the development and maintenance of all animals. And so the fact that this is essential in development, so apoptosis can be a good thing. And I want to show you a picture here that shows the example of apoptosis being part of normal development. This is a picture of the development of the paw in a mouse. Early on in development, there's tissues between the digits, and over time, that tissue disappears, so you have digits. And the tissue disappears because the tissue between the digits undergoes apoptosis, and they actually kill themselves off. This happens in the development of our fingers and toes as well. So early on in development, we have tissues between our fingers and our toes, and as a normal process of development, those tissues go through self-suicide, apoptosis, and therefore we have free-moving fingers and toes. All right? All right, so the things that you picked up today, where it says chapter 45, it's just a small section of chapter 45. I want to pull in with cell signaling. I'm going to teach you specific some specific hormones in our body and what they do and how they communicate, and therefore regulate certain things in our cells. So it's just a small section of chapter 45. I am going to make a small little video probably tomorrow morning for that. All right, so I'll send a remind text out when it's done. All right, and then the other two things are packets, much like the other packets as well that we've gotten with the information from chapter 45. All right, so and our test is going to be on Wednesday.